VariantGrid is a web app to store and analyze variants. Run it on your own server to scan your disks. QC graphs show how this run, in red, compares to others. Or just upload your VCFs to VariantGrid.com. Here's some fake patient data. We import plain text from our LIMS and fuzzy match phenotypes shown in orange, OMIM in blue, and genes in green. This allows us to search through patients that match a phenotype or disease. Now let's look at a single variant. Variants are annotated with over 100 columns. At the bottom are samples that have the variant. VariantGrid is your own population database with phenotype-genotype links. This family is from one of our lab's research projects with 1.8 million variants from whole exome sequencing. First, we'll open a VCF file and select the 3 samples. Now, we'll build the pedigree. The phenotypes were copy-pasted directly from the paper. The mother also has an intellectual disability, so we'll set her as affected as well. An analysis was created with 4 different inheritance models: recessive, de novo, dominant, and compound heterozygous. I'm adding a phenotype node. Connect the output at the bottom to the input at the top. We'll delete the recessive inheritance filters and move the other ones over. You can edit the filters in real time. Remember, this is 3 exomes with 1.8 million variants. Clicking on a node shows a grid of variants and the current filter. You can click on one of the subcounts. This one shows just the ClinVar pathological variants for this node. You can jump around and compare different filtering strategies. Here we're changing a filter, which recalculates everything downstream. Annotations change over time, and VariantGrid stores all of these different versions. This allows you to track changes over time. For instance, this variant had no ClinVar entry in March, but was listed as pathological a few months later. Tracking changes over time is key to automatically reanalyzing unsolved cases.